Yesterday, I included some slides in the hands-on folder under, I think it's experimental design, so on the box. So you might want to grab those. It's a RMD file in this PDF, if you haven't already got them. So I'm going to go through this on a PDF. What I suggest is that you open R and open the Markdown file, and you can go through and run the code as we go. You should be able to just click the knit button and reproduce this document with a different date. So I'm going to talk about a few concepts that Soledad covered, and we're going to do some simulations to illustrate them. And so I guess I'll get started. We have 25 minutes. So I'm going to start with multiple testing, and we're going to do a simulation and show that if we don't account for this, we can have really bad outcomes, conclusions. And then I'm going to talk about batch effects and experimental design. And then we're going to talk about power when batch effects are present. So I've output the code as I knitted this, so the code will be in the document. So these are three packages. These are three packages that were included with the package list that you had. And so we're just loading those, setting the seed. And so the objective, as I mentioned, is to explore multiple testing, batch effects, and batch effects on power. So the idea is if you test many, many variables and you don't adjust for multiple comparisons, and you just look at the raw p-values, you're going to get things that look significant and may look interesting to you. But you might follow them up with a lot of money and lab experiments and things like that, and you'll just be wasting your money because they're just false positives that we expect. So what we're going to do is we're going to simulate data. So we're going to simulate data for 10,000 genes, and it's just a random generation of sampled from a normal distribution. And then we're going to randomly give each sample either 1 or 0, depending, like treated versus untreated, or blood versus urine, or something that we might be interested in. So we're going to do 20 total samples, so 10 per group. So this is just the parameters to set up the simulation. And then using those parameters, this code will generate, so this is our group variables, so treated versus untreated, for instance. Each sample gets a 1 or a 0 with a 0.5 probability. And then we're going to generate the gene expression. So in var up here is a number of variables, so that's 10,000. So if you're running this again later, you can change any of these to make it specific to something you're interested in. So we're doing this 10,000 times, and we're just generating variables from a random normal. So these variables are generated independently from the treatment, so we don't expect any true associations. So then we're going to just test between the two groups. So we have zeros versus ones here, treated versus untreated. And we're going to test each of these 10,000 variables for an association with this treatment variable. And we're going to use this multi-t-test function that Dr. Coombs wrote because it's fast. So this function runs a t-test for 10,000 variables. It's very fast. You get the results. And then from that, I'm pulling out the p-values. And so now we have a vector of p-values. And so I'm just naming, adding a name here. So remember, these are randomly generated with respect to treatment, so we don't expect any true, or we know there shouldn't be any true associations. And we're going to take a look at them. So this is head p-value. So this is our vector p-values. I'm looking at the top 10. And you can see these are pretty significant. These are very small p-values. And these are randomly generated again. Keep that in mind. There's no reason for these to be associated with a trait. It's that we generate so many that some of them, by chance, are going to be able to separate or be different between our treatment groups. And so if we look at the histogram, this looks very similar to what Soledad showed. It might be the same. It might be the same one. And you can see at 0.05, as Soledad mentioned, we have, if we just use the raw p-value of 0.05, we're going to have 500 significant results. These are all false positives. So if you run this experiment and now you say, oh, we have 500 genes, we're going to throw them into IPA, which you'll hear about later. Some pathways are going to come out. You're going to think they're very interesting. You're going to design lab experiments, and they're really not meaningful. They're just, it's an illusion. So earlier, I don't think I mentioned it, I decided to use a 0.01 cutoff just because a p-value threshold is, it's not really, there's no defined reason to select 0.05 or 0.01. So if I use a 0.01 cutoff, there's 89 genes significant here at 0.01. Again, these are randomly generated. So the p-values are small. Some reach our predefined significance level. So now I'm going to take these 89 genes, and I'm going to try to cluster our samples and see if the two groups that are two treatment groups come out. So I'm pulling out the significant genes. So the p-value is less than p cut, which is 0.01. And then I'm going to cluster them with Pretty Heat Map, which is kind of a shortcut heat map program that has some nice settings to make it easier to make heat maps. So this is the heat map that we get. So what you can see is you have this group of genes that is apparently upregulated in group 0. This group of genes that is apparently upregulated in group 1, and they can perfectly separate groups. So we can perfectly separate this randomly generated treatment variable, or these samples, simulated samples, by a treatment variable with these randomly generated genes. And again, these genes are generated without any knowledge of the treatment categories. So the reason I'm pointing this out is I've heard many times, like, so I'll be working with an investigator, and they won't want to adjust for multiple comparisons because nothing's significant, and which is not a good reason not to adjust. And so they'll say, well, let's take the top genes and look if it clusters, and they'll see something like this. Oh, well, it clusters well. So there must be some meaning to it, but there's no meaning. These are just random generated vectors. So the reason they're clustering is because we selected the ones that separate the groups well to begin with. So we expect this to happen. So this is not adding any information to the importance of those genes. Again, these are randomly generated vectors. So as Soledad mentioned, we can adjust for multiple comparisons. And one way is a Benjaminian Hochberg. And this is a simple code. The command is padjust, and if you look at the help in R on this, there's several methods you could use, and I'm using FDR, which is one of two ways to specify Benjaminian Hochberg. And you see now, if I look at the top p-values here, the top ones or these are Q values, false discovery rates, if we assume that this gene is significant. They're at 0.27. So now it's much less interesting, right? So we've adjusted the p-values. Now it's what we expect. Nothing is really significant at a level that would be of interest to us. So this is the reason why you need, it's very important to make sure you correct for multiple comparisons. So in conclusion on this section, if we're running many tests, we have to adjust the p-values just because we expect to get false positives by chance alone. And clustering the samples based on the significant p-value, significant genes does not add any information to to the validity of the genes being significant or being true associations, because there were no true associations here. So questions on that one? I have 2 more sections, yes? So the Benjamini and Hochberg, it's a FOSS discovery rate, and so we're saying if we declare this p-value significant, this is our significance threshold at the p-value of this gene, how many of our total significant results that we declare significance at this threshold, what % do we expect to be FOSS discoveries, to be FOSS positives?
Do you mind elaborating more on what you said about these genes not being significant if they're clustering well?
So first you see that we randomly generate just vectors of numbers that I'm calling genes, and they're not generated with respect to the treatment variable, which is just 0 and 1. It's all random. So we know there's no true associations here. So we can still there's still going to be some that are randomly associated with that variable if we generate enough of them, just by chance. And if we pull out only those and try to cluster the samples based on those, we can cluster the samples well because we pick genes that by random chance are associated with the difference between the 2 samples, or can split the 2 samples. Is that?
That's because we didn't do multiple comparisons.
Well, so if we did multiple comparison, we wouldn't have selected any genes probably, right? We would have said we have nothing of interest here. But always if you pre-select based on association, or not always, but in most cases, you can cluster samples well based on the category or the variable that you tested on, just because that's how they're selected.
So how do we test the true association?
Well, we never know if it's a true association. What we can do is use, so we can adjust for multiple comparisons and use a threshold where we know, for instance, the FDR, if we use this threshold, we expect 90%. So if we use 0.1, we expect 90% of our declared true or declared significant values to be true positives. And you can use that to inform, let's say, so if you're doing a massive experiment with lots of genes, you may say, we're willing for 10% of our results to not validate in the lab to get the 90% that do. But you never know for sure without, you can get close with functional validation and things like that, but I don't know if there's ever a way to say that you know for sure. All right, so I'll move on. So the next thing we're going to talk about are batch effects, and this is another issue that has come up many times with investigators that I've worked with, and Soledad mentioned it as well. If you don't consider batches in your experimental design, you could end up with a very expensive experiment that you can get no good results out of or no believable results. And so again, we're going to simulate data. And this first thing here is just a function to plot multiple curves. It's kind of once I define this, it's kind of a shortcut instead of adding line by line. So again, simulation parameters, setting the seed. Here we're using an N of 50. This is the mean of batch 1, mean of batch 2, standard deviation of batch, mean of genotype 1 or group 1, mean of group 2, standard deviation of group. And so I'm calling the seed there, and now I'm setting the seed. So the way I'm going to go through this is I'm going to do a well-designed experiment, the worst possible designed experiment, and then a not horribly designed experiment. Soledad mentioned all 3 of these possibilities. So first we're making the data. So we're making batch 1, batch 2, and these are group 1, group 2. So batch 1, batch 2 have group 1. Batch 1, batch 2 have group 2. So, and then I'm making these 4 data frames and then just putting them all together so I have one data set with 2 batches, and 2 groups. Now I'm going to add the group effect, which are based on those initial parameters that we set. So here I'm simulating an effect. So the previous simulation, there was no effect simulated. But here I'm actually saying there's a difference in effect between, there's a difference in expression between batch 1 and batch 2. There's a difference in expression between group 1 and group 2. So I'm simulating this. So I don't know if we call this a true effect, a simulated true effect. So we've simulated the data, we've simulated the expression, and we're looking at the expression of one gene here. And now we're going to just take a look at the data. So you can see the way I simulate it, now we have equal numbers in batch 1, group 1, batch 1, group 2, batch 2, group 1, batch 2, group 2. So this is the optimal design. We're balanced. We've got both groups represented in both batches. And so these batches, by the way, could be chips, for instance. If you have a bunch of samples on a chip and you have 2 chips, now we have 2 batches. And there are chip effects. So we want to make sure that our group is not confounded with chip, like all of group 1 on chip 1, all of group 2 on chip 2. So and this is the good design, the best design here. And so here we can look at we're looking at just the histogram of the data across everything. We can look at the box plots. So here we have batch 1, group 1, batch 2, group 1, batch 1, group 2, batch 2, group 2. And you can see looking at, so group 1 versus group 2 in batch 1, for instance, so it's this one versus this one, you can see there's a small effective group. But if you look at the batch 1 versus batch 2 in the same group, you can see the batch effect is pretty large as I've simulated it here. And here I'm just plotting the densities by batch, so this is the distribution, so you can see batch 1, batch 2, there's a large effect. And then the same thing by group. And you can maybe see there's a little bit of a mean difference here, but you can definitely see the batches here in each group. So now we have this data, and we're going to test for an association. So the first thing we're going to do is ignore batch in our model. So we've generated this data. There are batch effects or group effects. And I'm just running a linear model. So that's this call, and the summary gives us this output. And you can see here that the group, if we're going to use 0.05 as a hard threshold, which is probably not a good idea, but for discussion's sake, it's not significant. So you see so this is the p-value here. So the group effect, our test, in most cases we'd say, well, we don't have enough evidence to reject the null of no effect. So one thing we can do is pre-adjust the data for batch. So we're basically removing the mean adjusting by the mean of each batch, so the batch effect no longer exists. So to do this, I'm just running a linear model and taking the residuals. The linear model just has an effect for batch. And so now we have adjusted expression. So this is the expression data adjusted for batch. And so after we adjust it, now we can run the linear model with the adjusted expression and analyze and see if we have a group difference. And here now we see that this is quite significant. So the batch was adding so much variation that we couldn't really detect the group effect. But once we remove that, now we can detect the group effect. Yes. So I'm just using a linear model and taking the residuals, but you can just think of it as adjusting by the difference in the means of the batch. So the batches have 2 different means, right? And so we can get rid of those. Yeah, so you can think of this method as like a normalization, yeah. I'm going to show a different method next, and this is just using a multivariable model. You'll see the results are very similar. So here I'm just running one model with batch and group. And again, you can see the p-value is almost identical. And I didn't look at the estimate, but the estimate's very similar too, maybe exact. So then I just look after the pre-adjusting for batch, look at the distribution. And still a little bit of a dip here, but you can see that the means, it's easier to see a difference between those 2 curves now than when you had the 2 humps and they were all spread out. So now I'm going to go to the poorly designed experiment. So again, I'm making a data frame. What I'm doing is putting all of group 1 in batch 1, all of group 2 in batch 2. And then I'm going to add the group effect. And then we're going to look at the data. So here we have 100 patients, or 100 samples in batch 1, group 1, 100 in batch 2, group 2. So you can see that there's complete confounding here. That there's no batch 2, group 1, no batch 1, group 2. And we can look at the distributions again. In this plot, you can see these don't exist. And you only have the difference between batch 1, group 1, batch 2, group 2. And we can plot the densities by batch. We can plot the densities by group. What you'll notice is that these are identical because batch and group are the same thing because we've confounded them. So it's like if you have all of your samples treated one way on one chip and all of your samples treated another way on another chip. Or if you took some samples that had one type of a disease and you run them and sequence them and then 6 months later decided you want to take a different subset of that disease and run those. That's an actual example. So then we can test for association, and I'm going to do the same thing. So we're testing the unadjusted. And you can see group is highly significant. So we're not accounting for batch, but this is because the group is also picking up the batch effect because we can't separate them from group. If we do the same thing with the pre-adjusting, you can see that once you pre-adjust for batch, there's no effect for group because you've also pre-adjusted for group because they're completely confounded. And in the last case, in the multivariable model, you can't even estimate the group effect because batch is accounting for all the variation of group because they're the same thing. You basically have two variables that are the same thing in the model. So this is a design that's not rescuable. In the end, if someone brings this data, we can't separate out what is an effect by group and what is an effect by batch. So I show the density here, and you can see once we adjust for batch, the means are approximately the same. It should be exactly the same. So the last case I'm going to show is completely at random, and this is another example that Soledad mentioned. Simulate the data, add the group effect. We're going to explore the data. Now here, since we did it completely at random, we didn't design it to be balanced. We can see we at least have all of the quadrants represented here, but they're not all 50. So we can look at the histogram, look at the effects. This is very similar to the optimal design. The densities again, and then testing for association. We see there's no association so this is without adjusting or accounting for batch. If we account for batch, so the pre-adjusted, you can see that we're trending towards maybe something. And then a multivariable model will be the same. So if you can design the experiment from the beginning and randomize the samples, that's what you need to do, and try to balance the design. And simple randomization is better than none, but it's not necessarily optimal. Maybe I hit all these already. So I know I'm going quickly, but I'm probably going to go even more quickly. Any questions on that first? So now we're going to look at batch effects and power. And so batch effects can, if you don't design the experiment well, batch effects can really be a problem. If the experiment's well designed, you can increase the power, as we've kind of hinted at above, by including the batch effects. So again, we're going to generate the data, simulate the data. And in this case, I have a parameter. So what I'm changing is the effect. So I'm going to use multiple effect sizes and see how well we can detect them. But this star J can be moved up here or down here, and then you change this to whichever you want to change, so overall mean. And this is just a multiplier, so this is 3 times 1, 3 times 2, 3 times 3, and these are the effects that are being simulated. This is calculating the mean of batch 1, batch 2, group 1, group 2, and then we loop this. And this is the number of reps, and I think I'm doing 100 here. So this is all looped by the number of values. So we're going to have 5 levels of group effect. Sorry, I know this is hard to follow with me going back and forth. And then the number of reps. So we're going to do this 100 times for each effect. And in the end, what we're going to get is each effect size is going to have 100 results, and we're going to look at how many of them are significant. And we're going to do this by a linear model with adjusting for batch, and a linear model not adjusting for batch. So this is batch adjusted expression. So then we're going to tally the power. So now in the end, what we're going to end up with is an object called p-tally, and what it has is the p-values and the effect size that we've simulated. And we're just going to tally this to figure out how many of them were significant in that test. And so that's what I'm doing here. I'm getting the percentage, or I'm counting the number that were significant, and then I'll get the percentage for each batch adjusted and non-batch adjusted. And now we're going to look at the results. So this is all just plotting the results that we just generated. And you can see, so this is effect size, so this would be 3, 6, 9, 12, 15. And you can see when we batch adjust, it increases the power that we have in the analysis. So this is, again, the well-designed study. And if we design it well, we can remove this batch effect or the variance associated with batch, and that gives us more power to find like the group differences, what we're actually looking for. And so that's the difference in these curves. So this is the unadjusted, and you can see the power, which is on this axis, is much lower than if we batch adjust. One conclusion is that one way to determine power or the numbers you need is through simulation. And if it's a really complicated experiment, sometimes this is the only way to do it. And as you kind of saw in the first batch section, by accounting for batch, we can increase our power to do the analyses.